Hi there, I'm Chris Mintoff, one of the lab managers here at Peter MacCallum Cancer Centre in Melbourne, Australia. Happy National Science Week to everyone and thanks for tuning in. Here at Peter Mac we have over 700 scientists, all working on research into lots of different types of cancer. There's more than 100 types of cancer and we're looking to understand them better and develop new treatments. I'm really excited to show you behind the scenes and walk through the lab space and hopefully meet some interesting and amazing and passionate scientists here at Peter MacCallum Cancer Centre. We're just here in the open space, in the lab space, and we're just going to approach our Zebrafish facility and see if Vicky Tan, one of our scientists in here, can actually show us around. Hi Vicky, how are you?
Very good, thank you.
Can you talk to us a bit more about what you currently do in this space and your research?
Yeah, definitely. So I'm currently a second year PhD student here at Peter Mac, and my project primarily focuses on liver regeneration and also studying liver disease. We use zebrafish as our animal model here. And the great thing about using zebrafish to study liver disease is that we can actually have a fully functional zebrafish liver from about 4 days, which makes us be able to do experiments from a really early time point. So, for example, we can study liver development and its response to drugs such as Panadol, all within 7 days of the zebrafish being born. So, yeah, it's pretty cool.
What are the advantages of using the zebrafish as a model to study cancer?
Yeah, so that's a great question. Zebrafish are great because a lot of the genes that we see in humans are actually conserved between zebrafish and humans. So there are about 70% of genes in the zebrafish that we also see in humans. And more importantly, a lot of the disease genes are also conserved, which makes it very easy to use zebrafish to study the same genetic and also physiology that we see in human diseases as well. And one of the other cool things is with a given breeding pair, so a male and female, we can actually breed hundreds of embryos overnight. And this allows us to have a lot of animals to work with, for example, for drug screens, identifying what kind of drugs that these fish are responsive to at a very large and cheap scale as well. So one of the main experiments that I do each week is a regeneration assay. So in our zebrafish, the hepatocytes, which are the main cells of the liver, express a fluorescent protein, GFP. So what this allows us to actually do is see the hepatocytes in these zebrafish light up green. Because the cool thing about zebrafish embryos is that they're transparent for the first 7 days of their life. So we can actually image these fish based on the GFP expression and the green colour of their liver and kind of track how the liver regenerates following 3, 4 or 5 days of liver injury. So yeah, that makes us be able to do in vivo imaging. So what that means is actually image these animals while they're alive rather than having to kill the animals or also kind of have static time points. So it's a really cool way to track liver growth and development.
So I'm just standing outside the Centre for Advanced Histology and Microscopy, aka CAM, and yeah, let's just walk in through and see who we can see. And here we have Paul Macmillan, he's the head of CAM. Paul, can you kind of tell us a bit about what you currently provide for our scientists here at Peter Mac and how this facility works?
Yeah, so here at CAM we provide research support for researchers to bring in their samples into the core. We can help them process them and make them ready to image on high-end microscopes that we have within the facility.
Okay, so here we're currently in one of the rooms in the CAM facility. This is one of our clinician researchers, Kazmira Wilson. And Kazmira, hi. Can you talk to us a bit about your research here at Peter Mac?
Yeah, sure. So what I'm using here is one of the light microscopes, and this microscope has the capability to regulate temperature as well as carbon dioxide concentrations to maintain cells that we put into it at the conditions that we need to. And then we can run this microscope for lengthy periods of time. So for this experiment, I've been running this for 48 hours now. And what I'm doing is imaging cells in real time over that 48 hours. So if you like, I can show you what I'm doing with the cells and explain a little bit about it.
Absolutely, that would be great. Thank you.
So what we do is we actually take biopsies from patients that come into the operating theatre here with rectal cancer. And when we take those biopsies upstairs, we actually grow mini tumours from them, and we call these tumoroids. And here you can see these are all the mini tumours that we've grown from the patient biopsies. And at the same time as we grow those tumours, we also take some immune cells from this patient. And this is what you can see, all these little dots here. These are some lymphocytes, some immune cells that we've also isolated from this patient. And once we've grown up the tumours and the immune cells, we pop them in a special plate and bring them down to this microscope, and we look at them with images every 2 hours for 48 hours. And what we want to know is, can these immune cells kill these little mini tumours? Because we know that the immune response is very important when we're looking at the ability to kill the cancer cells. You can see when all this red goes off, that's what we use to look at cell death. So you can see these immune cells are coming in and they're actually killing the little mini tumours, and then they're releasing the fluorescence or the red colour, which indicates that they're dying. And so what we're trying to use this experiment for is to work out why some patients will have a really good response to some chemotherapy and radiation therapy, and the tumour will essentially shrink away to nothing, and yet other patients don't have a very good response at all. And we're trying to work out if there's a way to predict which patients would have a good response and which patients wouldn't, so then we can personalise their treatment.
So I'm here with Alicia. Hi, Alicia. Alicia is one of our scientists here within the CAM facility, and we're standing next to this big piece of equipment. Can you just tell us a bit about what this equipment is and how you currently use it in your day-to-day work?
Yeah, of course. So this is a transmission electron microscope. Basically, to use this equipment, we take some tissue or cells, embed them into really small blocks of resin, cut them so that they're only 1 millimetre by 1 millimetre square, and that makes it so that they're thin enough that an electron beam can pass through them. Why we want to do this is so that we can see inside the cells. So researchers might be looking at nerves and looking at the sheath that's around nerves and how thick or thin it is. They might want to look at organelles in the cells like the nucleus or the mitochondria and see if what their research that is, what they've been doing to the cells or tissue is affecting it internally. So this is a level that you can't see at light microscopy. Normally you wouldn't be able to see the nucleus in this much detail. You wouldn't be able to see organelles. So yeah.
Awesome. And can you tell us a bit about what the difference between this type of microscope and normal light microscopy is in terms of magnification and imaging?
Yeah, so instead of using light microscopy, it uses lasers and photons, so light that you can see. This microscope uses an electron beam, so it's not something you can actually see with the naked eye. And instead of sort of the resolution of light microscopy, we can technically go up to about 250,000 times magnification. Realistically, most people image at 5 or 10,000 times, but that basically lets us be able to see a whole mitochondria in complete detail. We can see all of the membranes and all of the detail of it, which is something you wouldn't really be able to see in light microscopy. You just see a little coloured blob.
Fantastic. I can see here you've got an image set up on the screen. Can you just briefly talk to us a bit about what we're actually seeing here?
Yeah, absolutely. So this is a T cell. What we're seeing is the entire cell. So you can see that it's only about 4 to 6 microns big. This big blacker blob in the middle is the nucleus. And all these little smaller ones you can see are the mitochondria that I've been talking about. And you can actually see sort of the membranes inside them as well. All of the darker material you can see in the nucleus is the DNA. So that's pretty cool.
Yeah, well, fantastic. Thanks so much for your time.
That's okay.
So here we are just outside the Victorian Centre for Functional Genomics. This is yet again one of the few core facilities we have here at the Peter MacCallum Cancer Centre. So yeah, let's just walk through and see if we can get a virtual tour. So this is Twishi, one of our scientists here at the VCFG. Hi, can you just briefly talk a bit about what you currently do here at the VCFG?
Sure. So we are a core facility at Peter Mac. VCFG stands for Victorian Centre for Functional Genomics. What that essentially means is that we look at changes that happen in the genes, which are a part of your whole genome, which is the DNA, which is a part of the DNA of your cell. And we change the function of the genes in various ways. And then we see what happens to the cancer cells, in particular, for example, when you alter or change the expression of the genes in them. Do they start proliferating more? Do they change their shape? Or do they start growing slower? And that can be a way that you can develop drugs against and kill the cells.
Awesome. And what's this room currently used for?
So this is the tissue culture room, which basically means this is where we grow cancer cells. We grow them, expand them, and play with them, for the lack of better description, in these hoods over here. And then they are allowed to grow and prosper in an environment that is conducive or good for their growth, which is at 5% CO2 levels and 37 degrees, just like our bodies. And that's done in the incubators here. So I'll just quickly show you what's in here. So the cancer cells are grown in different size plates, or they're not petri dishes anymore like back in the old day. They're flasks and they look like this. And here millions of cells are growing as we speak. And so from here, as a facility, because we do a lot of drug screening as one of the things we do, so I was thinking of taking you through the process of how that's done in our lab. Along with drug screening, we also edit genes using drugs, but also by modifying them with this new technology called CRISPR. And there are a lot of different ways we do this in the lab.
So I understand that this is one of the many rooms within this facility. So I was wondering if we can also look at some other rooms where I know there's some really big and expensive equipment that you use.
So let's go to the next room, which we'll follow through.
Awesome.
So this is one of the equipment we have here. It's called a biotech. We use this to put those cells that were growing in a flask into something this small. It's called a 384 well plate. So it literally has 384 wells. And so the millions of cells, putting them in each of these wells, we can do using this machine, and it does it in less than 5 minutes on multiple plates in one go. So if you're doing a drug screen where you want to look at 1,000 drugs in one go, what they're doing to your cancer cells, you put the machine on. Using this machine, you put the cells into 30 of these plates in half an hour. And so once we're done doing that, we let the cells settle onto the plate overnight, and then we put drugs on them. And to do that, especially on a plate this small and the size this small, we do it using a robot in that room, which we're going to go to, and Henry's going to talk you through the process there.
Perfect. Thanks so much. Hi, Henry. This is Henry, one of the scientists working at the VCFG. I can see some robotics here. Can you talk a bit about what this equipment actually does?
Sure. So the idea of the robotics is we're wanting to automate everything. So at the moment, this is the kind of thing we'd be using normally right in the lab. So this is a single channel. It can do one thing at a time. You can upgrade to something like this, which does 8 things at a time, 8 different wells. But we have 384 well plates, and we have multiple of those plates. And so that's when robotics comes in. So the idea here is those cell plates, which Twishi was talking about before, are put on the stage, and then they're stamped. So the liquid is moved from the compound plate or the drug plate into the cell plate, and that's done automatically. So the idea is it's automated, so it's easier for us. We don't want to be doing this by hand using these. It'd take a long time. It's also easier for low volumes. So we're dealing with really, really small volumes of liquid, and the robot does all of that for us. So it's more accurate as well.
Yeah, that's fantastic. So obviously that saves a lot of time for you to actually pipette so many samples, and also reduces human error, right? So
Yes,
that's
exactly.
fantastic.
That's the one.
Awesome. Thank you. So what happens after finishing a drug screen?
So when we finish a massive drug screen, we normally fix the cells, which basically means we treat them with a chemical that makes them stick to the plate so they don't come off. And then we color them with different colors, trying to look at what's in different parts of the cell. So we'll have a different color to look at the cytoplasm of the cell versus the nucleus. And then we put them on this machine, which might not look like much, but it's actually a microscope. It's a big difference from the old school microscopes that you are used to looking at, where you look down the binoculars. And this particular microscope has a orbiter attached to it, which is currently moving a plate from a standing point to place it in the microscope so it can start imaging a plate. Again, this is all automated, so this will happen continually till all the plates in stack 1 are finished scanning, and then they get deposited in stack 2 when they're finished. And so this can happen all through the weekend. When you have 30-something plates, it just does it automatically once you set it up on the software.
Henry's here with a new piece of equipment. I was just wondering if you can tell us a bit about what this equipment actually does.
So this is called an IncuSite. It's quite a cool piece of equipment because it's a microscope that's actually housed inside an incubator. So this is the same incubator that was growing the cells before. And it's actually this piece of equipment here. Why it's cool is because the microscope can image the cells in real time. So that means every hour we take an image of the cells and we watch them grow over time. So those same plates you saw before are here. So we have some 384 well plates and some 96 well plates. And so there's cells in each of these wells. Each well has a different drug in it, for instance. And there's a microscope housed that takes images looking up here at the bottom of the plate. And that's quite a unique feature. Normally, it is the end of the assay that you would take images and you'd look at what's happening, whereas this is tracking it the whole way through. So that's why it's a neat piece of equipment.
That's fantastic. Yeah. Thank you. So, hi everyone, I'm just standing here. This is actually Professor Shireen Loy's lab space. They basically specialize in breast cancer. And we'll just walk through and hopefully speak to some people who work in this lab and see what they are up to. Hi, so this is Courtney Van Galen, one of the scientists here at the Peter MacCallum Cancer Center working in Shireen Loy's lab. I was just wondering, hi Courtney,
Hi.
I was just wondering if you can tell us a bit about your projects and what you currently do in this lab.
Yeah, so this lab focuses on really understanding all about breast cancer, so particularly the genetics of it and the immune system and how that interacts with breast cancer. And so at the moment I'm growing up some cancer cells and I'm going to see what happens when I treat them with a whole bunch of different drugs.
Okay, so can you just tell us a bit about what you're currently doing?
Yeah, so at the moment we've taken up some frozen cells that are from a cancer patient and we're just regrowing them in a sort of controlled setting where we can control what we give them in terms of media and all that yummy stuff that cells need to grow, and then I'm going to lift them up and put them in plates and test them with a whole bunch of different drugs to see if it kills the cancers or not like we want. So this is a centrifuge. If we open it up, you can see it's got lots of little spaces where we can put all the different sort of blood tubes in, and when we get things like patient blood that we want to look for specific types of cells, like immune cells in them, we can spin this, and as the centrifuge spins around and around like those little carnival rides when you were a kid, it'll separate all the different aspects of the blood into like the immune cells and your red blood cells and your white blood cells and the plasma, and we can then extract those specific aspects of it and do a whole bunch of different tests on them depending on what we're looking for. When a patient has breast cancer and it gets removed through surgery, chunks of it get sent for various testing where they stain it so that we can identify kind of the shapes of the different features that make up the breast cancer itself. And we can look at it on a larger scale on the computer screen, and you can see that there are all these big bits of cancer in here that look quite messy, and all these little cells here, the dark purple stains, they're called TILs, which are tumor-infiltrating lymphocytes, which are basically the immune system learning how to attack the cancer. And so we've got a website through our lab that teaches you how to read these TILs and score them, because essentially the more immune system cells you have in your cancer, the better your outcome is. So patients that have 50% of immune cells, like in this case, are more likely to have a better response than those that have 10.